Good afternoon and welcome to the first in the Veramed free webinar series. Today we have Nick Cowans on the line, who's going to be presenting on the statistical input into improving data quality. So without further ado, I will pass over to Nick. Thanks, Nick.
Okay, thank you very much. Okay, so today I'm going to talk about data cleaning in a clinical trial and how it can really benefit from the involvement of statisticians. I'm going to draw on my experience of working in a large phase 3 respiratory trial, and I'm going to use examples of exacerbations and FEV1 endpoints, both of which I will explain more about later, to show how we as statisticians in this trial created novel visual tools to benefit data cleaning. So before I start talking about cleaning data in a clinical trial, I asked myself the question of what exactly are we trying to do when we are setting up and running clinical trials. And I thought, and I found out that clinical trials are trying to answer a set of questions, usually about the intervention of a drug. For example, a typical question you might ask when you're setting up a clinical trial is, does the drug I'm testing have an effect on survival? I.e., can patients taking this drug, can their chances of living longer be extended? Another common question in the respiratory therapeutic area is, can the drug slow down the inevitable rate of decline that subjects with certain diseases, respiratory diseases, have in FEV1, which I'll explain a bit more about later. And another common question in clinical trials in the respiratory setting is, can this drug we're testing reduce the number of exacerbations a subject has? And again, I will explain more about what an exacerbation is later on. But to reliably answer these questions that we're asking, you need to have confidence in the data you're working with, so that your statistical analyses have sound, reliable, good quality data behind them. And that's where data cleaning comes into clinical trials. So as I said, I'm going to draw on my experience of working on a large phase 3 clinical trial in the respiratory setting. And the study I worked on was very large. It had well over 10,000 subjects in it, and these subjects came from over 1,000 sites around the world in more than 40 countries around the world. So already, when you're thinking about data cleaning, issues come to mind of the scale of what you're sometimes working with in a large phase 3 clinical trial. When you also then look at the timeline involved, this is even clearer. So the study I worked on had a large recruitment phase. So the first subject's first visit on day 1 was followed by about a 3-year recruitment phase, where subjects were being entered into the study all the way through this time in different sites in different countries. Then once the last subject was recruited, this was an event-driven study, so we were then waiting for a number of events to happen to do with our primary endpoint. So that meant there was like another year, when all of the subjects were in the study and we were collecting data on them too. Then once we ended the study, we had about 6 further months when we were closing out sites. We'd stopped collecting new data about the subjects, but we were closing out sites and we were still doing data cleaning before we finally unblinded about 6 months after the study ended. So altogether, we had over 4 years of data collection occurring in our study, followed by about another 6 months before we unblinded our data, when we were still able to clean the data up. So data cleaning is an absolutely vital part of all clinical trials, because the data is being collected in lots of different sites, and we want to make sure that the data is as clean as possible. So usually the responsibility of data cleaning in a clinical trial will lie with a data management department. And the usual process is that a set of predefined rules is usually outlined before the study starts for things for data management to look out for with data issues. For example, all measurements should fall within some sort of sensible range, and if they don't, then they should be queried. So an example of this is, are all ages of the subjects in the study above 0 and below 100? And anything like that obviously could come from a data entry error. So when those sorts of rules are added to the live database that's being used in a clinical trial, data queries are sent off to try and clean the data. So especially when thinking about a large phase 3 study, you have to accept that you have a vast amount of data and you can't possibly clean absolutely everything. So although data management has all these rules set up and they are working to clean the data, they're usually based upon individual data points, and they are identifying things that are unusual about individual data points. However, normally in a clinical trial, you're not collecting data in isolated. It's normally you'll have a subject who'll come in for several visits, and you will collect data at each visit. So really you want to start thinking a bit more longitudinally about your data and not just thinking about an individual point and whether that point is unusual. You might want to think about data collected before and after and see whether that point falls out of what you'd expect from that. Another issue with data management cleaning your data is that they might not know when the study is set up exactly what the statistical analysis that will be done on the data will be, because sometimes that's not finalized until later on in the study. So as a statistician, you can obviously have more of a input into what data might need to be cleaned from a stats endpoint. So this slide just shows a typical data collection process during a study, and it's based on the study I was involved in. So for each subject in your study, various measurements are taken throughout the study to do with all your different endpoints and all your different logs, and the site will then enter the recorded information from those measurements into a live database. So this is already a possible source of error, because it could be a data entry error, because usually a measurement will be entered from a handwritten record or maybe a printout from a machine. So the site's entering that all the time into a big live database. So in the study I worked at, we actually received monthly extracts of the data throughout that 4-year period that we were where the study was ongoing. So obviously the data was blinded. We didn't know what treatment anyone was on, and nor did the CRO who were involved in the data entry or any of the investigators. So every month that data management CRO would take a snapshot of the database and extract it and convert it into SDTMs, which they sent to us, the stats and programming CRO, and we would then derive our Adams from that, and using dummy randomization, we'd start to build our final TFLs using the dummy randomization. And this meant that every month we were getting a really good look at the data, and we could see from our summary tables any odd points, so we could send out data checks based on whenever we saw minimums or maximums in tables that didn't look right. But it also meant that we could start to get a really good feel for the way the data was affecting our analyses that we had in our planned stats plan. So we started as statisticians making a lot of our own data checks, 2 of which I'm going to talk about today. And part of that process was working with the clinical team, and they were reviewing our data checks, and we were sending all of this information back to the data management team, who would then send them out as queries to sites. So there's a long process here, and it involves an awful lot of data, especially when you think that this is occurring for all of your 10,000 plus subjects, at all of your up to 10-15 visits for all of the different endpoints you're collecting. There's a lot of data here. So how and why should stats get involved in data cleaning? Well, as I've already said, going back to the previous slide, stats sit closer to the end of this process, meaning that they are closer to the final outputs that we're going to be looking at once the data is unblinded. So compared to data management, who are further up and who are looking at the data being entered into the database, stats have more of an idea of how the unclean data may affect the analysis of the endpoints that we're looking at. Also, because we're statisticians and we're using SAS and other programming languages, we can actually create novel visual tools that can show to clinicians and also the data management team and the sites visually how unusual data is going to affect our analyses. And we found that by making these tools, we ended up building a good close relationship with both our clinical and data management colleagues, which meant that everyone got a really good feel for the importance of cleaning the data with the stats endpoints in mind. So I'm now going to talk about two examples of endpoints that we as statisticians cleaned up during the study that I worked on. And the first one is looking at exacerbations. So exacerbations are a common respiratory endpoint, and the typical definition, and the one that we had in our study, was it's an adverse event. It's a protocol-defined definition. It's an adverse event of worsening symptoms of the disease that required treatment with either antibiotics or steroids. And in our analysis, we were doing both rates and time to first analyses. So we were looking at whether the drug we were testing had an effect on the number of events, exacerbations subjects were having, and also whether or not it had an effect on the time to the first, on average, the time to the first event that subjects were having on each arm. So that's the analysis we were doing. The way we actually collected the information was we obviously had an adverse event page in the CRF, and for every adverse event that was entered into the database, a question came up saying, does this AE meet the definition, the protocol-defined definition of an exacerbation? And we were relying on the investigators ticking the box saying yes or no, this does or doesn't meet that definition, and that was how we were identifying our exacerbations. So we realized when we were looking at our data in our monthly extracts that we were getting quite a lot of cases where we were having what we called overlapping exacerbations. So there'd be two adverse events recorded at exactly the same time, both recorded as exacerbations. So when we looked at that, we found out that what was actually going on was that investigators were recording an adverse event with some sort of clinical term, for example bronchitis, but they were also recording another separate adverse event saying that it was an exacerbation of the disease COPD. And really, this was being recorded in our database as two events, when really it was just one event, a bronchitis that was an exacerbation of COPD. So we found that it was difficult to communicate that directly to sites just by saying it. So we created this tool that produced a profile of subjects' exacerbations, and it's what's shown on the screen now. So it had the subject, all of the exacerbations that are in question, just two in this case, and it said when the exacerbation started and ended, the duration of it, and then the reference ID for the AE. This was sent out to our clinical colleagues who looked at it and would write a query text to the site saying, explaining the issue at hand. And we found that this cleaned up a lot of exacerbations, so we were no longer falsely recording this one event as two events. Another thing we found was that often two exacerbations would occur but very close to each other in time, but they would be recorded as two events. And this was obviously going to affect our rates, because this could be considered clinically as one event, but it's been recorded as two by the investigator. And again, we found that by presenting it visually on a profile like this, we were able to explain to the sites what our issue was, and then we could ask them should this be considered as one exacerbation, or would you prefer it to keep it as it is and have it as 2 exacerbations? And obviously it came down to the investigators' decision on how they felt this was appropriate, but having it visually like this helped them make that decision. And we also included the drugs, the concomitant medications that were taken at the time to help the investigators and the clinical team make their decision on what way to go forward. A third issue we came up with in exacerbations was that we would have evidence of an exacerbation from a different part of the CRF, but we were actually missing the vital adverse event page with the exacerbation tick box on. So, for example, as our definition was that an exacerbation you had to be taking either an antibiotic or a steroid, we found that there were entries on the ConMeds page of CRF where an antibiotic or a steroid was being taken, and they'd answered yes to the question, was this medication taken for an exacerbation? However, when we went back to look at the AE page, there was no AE marked as an exacerbation. So we were actually aware from the data that the subject was having an exacerbation. However, we were missing the really important part of having it on the AE page. And this inconsistency of recording exacerbations meant that we were missing a lot of events. So when we identified this and sent all these queries out to the site, we found that a lot, once they'd realised that they just hadn't ticked that box on the page, or they hadn't maybe entered an AE, more AEs were entered, and we started to realise that we were getting more AEs of exacerbations in our study. And overall, the impact of cleaning up these exacerbations meant that if we hadn't cleaned them up, having those overlapping exacerbations still existing in our data set would have meant that we would have falsely increased our exacerbation rates, because a subject who's really only having 1 exacerbation would have been recorded as having 2. Also, had we not identified the missing exacerbations, we would have had the opposite effect. We would have falsely decreased our exacerbation rates, and that would have changed our analyses. And also, because we were not just doing rates, we were also looking at time to first. If we had missing exacerbations, and that was the only exacerbation that the subject had, it could change the time to first analyses. And because we were doing all of this data cleaning while the study was blinded to us, we were not interfering in we weren't creating any bias in what we were doing. We were treating all subjects the same, and we were identifying all these issues the same. So the second endpoint that we looked at as statisticians and data cleaning was FEV1. So FEV1 is a common respiratory endpoint, and it stands for the forced expiratory volume in 1 second, and that means the volume of air exhaled by a subject in the first second when they're performing a forced expiratory maneuver, which is into a specialist machine, and it's basically a measurement of lung function for the subject. And we used it in our analyses in several different ways. The first and most important way was we were looking at the rate of decline of FEV1 in our subjects. So subjects with certain respiratory diseases will naturally decline in their FEV1 over time. Their lung function just goes down over time. But we wanted to know whether the drug we were testing had an effect on slowing that down. So if you're taking this drug, does your rate of decline slow down over time? We did that using a random coefficients model, but we also had another analysis in our data set where we were looking at individual regression slopes. So there were already data management checks in place checking FEV1. So they were looking to see whether values fell within a specified range. And that was obviously an important part. So if for example, there was a 10-litre volume of lung, or a 100-litre volume of lung volume, then that would be something that would automatically get queried by the data management, because that could just be a typographical typo or a data entry error. And they also looked at whether there was a difference from the baseline, because each subject has their own baseline value, and it's dependent on a lot of things like progression of disease, gender, even region. And if they had a recording that was out by a certain amount from the baseline, that was automatically queried. However, we realised when the data was coming in during the ongoing study phase, when we were getting these extracts, that we needed to have some more statistical checks because these checks that data management doing weren't quite enough to really identify all the problems. So in our stats department, we created a FEV1 profile tool using SAS and Excel. And what it did was it exported data from SAS using all of the ADAM data sets we created, and it was able to get read into Excel, and each subject would have their own FEV1 profile displayed when you selected them in the Excel spreadsheet, and it would show all the visits they had and the data points that had been recorded for them. And we could then set up our own rules that didn't just look at individual data points, but could look at data points together. So we looked at standard deviations. Did this subject have a standard deviation greater than 800 mils? Did they have a change from baseline more than a certain amount? Or does a data point change from the previous one by a certain amount? So you can see this subject here has had all these visits, and then there's a data point here that stands out when you look at it on a graph. And it's a change, it's risen a flag here saying that it's a change from baseline over 750 mils. So we produced this spreadsheet. All of the ones which had flags in were then sent to clinical, who could then type in their comments here and decide whether or not they wanted these things queried. If they did want them queried, that would appear up here in this box, and we would then produce a PDF which we could then send to the site explaining, that this subject has these data points. One of them looks unusual because of whatever reason we'd identified, and asking them just to verify their source documents to check that the data point had been entered correctly. And then the site would either change the data point if they realised they had entered it incorrectly, or they'd reply saying why their data point was so low. So in summary, from my experience of working on this large phase 3 respiratory trial, we found that data cleaning really can benefit from the involvement of statisticians. The data can be cleaned up in a different way to just involving data management, and including statisticians in data cleaning means that everybody involved in the process is more aware of the statistical endpoints in mind and why certain parts of data needs cleaning. And of course, statisticians benefit from being involved in data cleaning because they know that they know what endpoints the endpoints are, what the statistical analysis is going to be, and which data points they need to focus on for cleaning. We found that in this respiratory trial, it was really useful to represent graphically to both clinical and data management what was going on, and I've shown the examples of exacerbations and FEV1 profiles for this. And we, although we can't know for certain, we are able to be confident that by cleaning up these endpoints and these data points, we have had a positive impact on the endpoints and the statistical analysis in our clinical trials. So thanks very much for listening, and I'm now happy to take any questions.